All right, today we're going to tackle a question that sounds really simple on the surface, but is actually one of the most profound questions in all of science. We're going to go on a tour, a journey to the very core of what it means to be alive.
So think about it. What's the real fundamental difference between a rock and a rabbit? Or a tree and a teaspoon? It's a huge question, right? It goes to the essence of existence itself. And the answer, well, it isn't something you can just spot with your naked eye. The secret, it lies in a tiny, bustling, and unbelievably complex unit, the cell. This is the basic building block that truly defines what it is to be alive.
So to get our heads around this world within us, here's the plan for our explainer. First, we'll uncover how this secret of life was discovered in the first place. Then, we'll explore the two major designs that cells follow. After that, we're going to take a tour inside a bustling cellular city. We'll also meet the critical border patrol that keeps it all safe. And to finish, we'll look at a really curious exception that kind of breaks all the rules. So, ready? Let's jump in.
Okay, our story starts way back, not in some fancy modern lab, but in the 17th century. This was a time of incredible scientific curiosity. The invention of the microscope literally opened up a whole new universe, one that was totally invisible to us before. And it was here that humanity got its first ever glimpse of the fundamental unit of life.
You know, what's really amazing is how this understanding was built, piece by piece over centuries. It all kicked off in 1665 with Robert Hooke. He looked at a thin slice of cork and saw these tiny, empty boxes that reminded him of monastery rooms, so he called them cells. But he was just seeing the outer walls. Just 9 years later, Anton van Leeuwenhoek became the first person to see actual living things, tiny little creatures swimming around in a drop of pond water. The plot thickened much later, in 1831, when Robert Brown spotted the nucleus, hinting that there was some kind of control center inside. And that brings us to the final game-changing piece of the puzzle from Rudolf Virchow in 1855.
And Virchow summed it all up with this incredibly powerful Latin phrase, omnis cellula e cellula. It means all cells come from cells. Now, this simple statement was absolutely revolutionary. It finally threw out that old idea that life could just, pop up out of nowhere from non-living stuff. No, life only comes from pre-existing life through the division of cells. This idea was the final piece of the puzzle, forming the basis of what we now call cell theory.
And so these three simple ideas became the foundation of all modern biology. 1, everything alive is made of cells. 2, the cell is the basic unit of life. And 3, all new cells come from old cells. It doesn't matter if it's a tiny bacterium or a huge blue whale, these rules apply. This theory literally defines life as we understand it.
Okay, so now that we know what the theory is, let's look at the cells themselves. As it turns out, life on Earth pretty much follows two main architectural plans, two basic blueprints for building organisms, the prokaryotes and the eukaryotes.
First up, we have the prokaryotes. These are the ancient ones, the originals. Think of them like an efficient one-room studio apartment. They're small, their DNA just kind of floats around in a region called the nucleoid, and they don't have all those fancy internal compartments or organelles. Bacteria are the classic example. And then you have the eukaryotes. These are the more complex cells that make up everything from plants and fungi to animals, like you and me. They're way bigger, and their defining feature is a proper nucleus with a membrane to house their DNA. Plus, they are jam-packed with specialized organelles, each one doing a specific job. Now, even within the eukaryotes, we see some cool specializations. Let's just quickly compare animal and plant cells. A plant cell has this rigid outer cell wall that gives it a fixed, almost boxy shape. It's what helps a plant stand tall. Animal cells? We don't have that, which allows our cells to be more flexible. Plants also have chloroplasts, these amazing organelles that let them make their own food from sunlight. We animals can't do that. We have to eat our food. And you'll almost always find a huge central vacuole in a plant cell, which is basically a giant water tank. Each of these differences perfectly reflects their unique way of life.
All right, to really get a feel for the eukaryotic cell, let's take that tour I promised. The best way to think about it is to imagine a massive, bustling, self-sufficient city. Every single part we're about to look at, every organelle, has a critical role to play in keeping this entire city running.
Our tour starts at City Hall, the nucleus. This is the absolute control center. It's got a double membrane for protection, and inside, it holds the master blueprint for the entire city, the DNA. From here, it sends out all the commands that control everything the cell does. And why does this matter?
Well, if that blueprint, the DNA, has any errors, it can lead to all sorts of genetic diseases. So keeping the nucleus safe and sound is absolutely vital.
Okay, every city needs power, right? That brings us to the mitochondria, the tireless power plants. What they do is take the food we eat and, using oxygen, break it down to generate something called ATP. ATP is the energy currency of the cell. It powers everything. And here's a mind-blowing fact. Mitochondria have their own separate DNA, which suggests they were once free-living bacteria that our ancestor cells swallowed up billions of years ago. They are literally ancient tenants living inside our cells, paying rent in the form of energy.
So the nucleus gives the orders, the mitochondria provide the power, but who actually builds everything? That's the job of the ribosomes, the city's countless little factories. They have one of the most important jobs of all, building proteins. They read the instructions sent out from the nucleus and assemble all the proteins the cell needs, everything from structural components to the enzymes that digest our food. They are the true workhorses of the cell.
Now, once all these proteins and other things are made, they need a way to get around. That's where the endoplasmic reticulum, or ER, comes in. It's this massive network of membranes, like a highway system that crisscrosses the whole cell. The part called the rough ER is covered in ribosomes, and it's where many proteins are made and modified. The smooth ER, which doesn't have ribosomes, is where lipids are made and, importantly, where detoxification happens. Your liver cells, for example, are packed with smooth ER to help break down toxins.
And finally, before these products are shipped out, they need to be sorted, packaged, and addressed. That's the job of the Golgi apparatus, the city's main post office. It receives proteins and lipids from the ER, puts the finishing touches on them, and then packages them up for delivery. Whether the package is going to another part of the cell or being sent outside the city entirely, the Golgi makes sure it gets to the right place. It's the final stop in a very organized assembly line.
So any well-run city needs a border, right? Something to protect it and control what comes in and what goes out. The cell is exactly the same. It has an incredibly smart and sophisticated border patrol that is absolutely essential for its survival. We call it the cell membrane.
The cell membrane is just a masterpiece of engineering. It's a flexible but strong barrier, but its most important quality is that it's selectively permeable. What does that mean? It means it's like a very picky bouncer at a club. It lets some things, like water, pass through easily, but it strictly controls who else gets in or out. This regulation is absolutely critical for keeping the inside of the cell stable and balanced, the perfect environment for life.
A perfect way to see this in action is a process called osmosis. Imagine you put a cell in really salty water. That salty water has a lower concentration of water molecules than the inside of the cell does. Since the membrane lets water pass freely, water will naturally move from where it's more concentrated, inside the cell, to where it's less concentrated, outside. The cell starts losing water, and as a result, it shrinks. This is exactly why your fingers get all wrinkly after a long swim in the sea. It's your skin cells losing water to the salt water.
So we've built this really neat and tidy picture of life. It's all based on the cell. But, biology loves to throw a curveball. Just when we think we've got it all figured out, we run into this strange and fascinating thing that seems to break all the rules we just established. I'm talking about the virus.
You see, viruses are the ultimate exception to cell theory. First of all, they aren't even made of cells. They're just a bit of genetic material, DNA or RNA, wrapped up in a protein coat. That's it. They have no cell membrane, no power plants, no factories. On their own, they can't do a single thing. They are totally inert, no more alive than a grain of sand. They only spring into something that looks like life when they infect a living cell and hijack its machinery to make more copies of themselves.
And that brings us to our final, and perhaps most interesting question. If our very definition of life is based on the cell, and viruses are not cells, if they're basically dead when they're outside a host, but can replicate inside one, what are they? Are they really alive? It's a question that pushes the very boundaries of biology and forces us to think about that blurry, gray line between life and non-life. How does life just grow? How does it fix itself when it gets damaged? And how does it keep going, generation after generation? Well, it's not magic. It's an incredibly organized, microscopic process, and we're about to break it all down. So, before we can understand how something grows, we gotta start small. I mean, really small. We have to get to know the hero of our story, the single cell. It's a pretty simple idea, but it's also incredibly powerful. When Robert Hooke first peeked at a slice of cork through a microscope, he saw these little boxes, cells. They are the absolute basic building blocks of everything alive. You, me, that plant on your windowsill, we're all just these complex, amazing communities of cells working together.
Now this quote here was a total game changer. Back in the 1800s, a scientist named Rudolf Virchow laid it out plain and simple. Cells don't just pop into existence out of nowhere. Every single new cell has to come from one that's already there. Which, of course, leads to the big question, how? How does one cell make another?
Well, the answer is this incredible process called cell division. It's where one parent cell just splits, creating new daughter cells. It's how life, at its most fundamental level, makes more of itself.
And this isn't some chaotic, random event. Oh no, it's a highly organized, step-by-step process we call the cell cycle. But here's the thing, not all cell division is the same. Living things actually use two main methods for two very different reasons. So let's start with the one that helps you grow and heal every single day.
First up, we've got mitosis. I want you to think of this as a biological photocopier. Its whole job, its entire purpose, is to make a perfect, exact replica of a cell. This is what you want for things like growing bigger or repairing tissue. So mitosis happens in what scientists call our somatic cells. That's just a fancy way of saying pretty much every cell in your body that isn't a reproductive cell. So we're talking skin, muscle, bone, you name it. And the end result? You get two new cells that are perfect genetic twins of the original. Same exact DNA, same number of chromosomes, a perfect copy.
And this whole copying process is like a beautifully choreographed dance that happens in 4 main acts. First up, in prophase, all the genetic material gets packed up tight. Then, in metaphase, it all lines up right down the middle, super neat. In anaphase, the identical copies get pulled apart to opposite ends. And finally, in telophase, the cell itself splits right in two. It's just incredible precision on a scale you can't even see.
And this is why mitosis is so unbelievably important. It's how you went from a single fertilized egg to the person you are today. It's how that cut on your finger heals up with new skin cells patching the hole. And in plants, it's what lets them just keep on growing their entire lives. It's the engine of growth and repair.
Okay, so making perfect copies is great for keeping you going. But what about making a whole new you? For that, life needs a totally different playbook. It can't just make a clone, it has to create variety. And that's where meiosis comes in. It's less like a photocopier, and a lot more like shuffling a deck of cards.
Here is the absolute key difference. Meiosis only happens in our reproductive cells, and instead of making 2 identical cells, it creates 4 totally unique cells, so sperm or eggs, and each one has exactly half the genetic information of the parent cell. Because it literally reduces the chromosome count, you'll often hear it called a reductional division.
So, how does it pull this off? How does it create unique cells with only half the DNA? Well, it's pretty clever. It doesn't just divide once, it divides twice. This 2-step dance is the key. It's what allows the cell to have the chromosome number, and through a really cool process where the genes get swapped around, it shuffles all the genetic information to create brand new one-of-a-kind combinations.
You can see why this is so huge, right? Without meiosis, sexual reproduction as we know it just wouldn't be possible. It makes the sperm and eggs we need, it makes sure every new baby has the right amount of DNA, and this is the big one. It creates all the genetic variety that allows species to change, to adapt, and to evolve over millions of years.
All right, so we've got these 2 powerhouse processes, both incredibly important, but also very, very different. Let's put them side by side so it's all crystal clear.
When you zoom way out and boil it all down, the difference is this simple. Mitosis is for growth and repair, and it makes identical cells. Meiosis is for reproduction, and it makes unique cells. One process builds and maintains the individual, and the other allows the entire species to continue.
And if we get into the nitty-gritty, the differences are even more obvious. Mitosis, that happens in your body cells. It produces 2 daughter cells, and they're identical with the full set of chromosomes. Meiosis, that's just for reproductive cells. It makes 4 daughter cells, they're all unique, and they each have just half the chromosomes. Fundamentally different jobs, fundamentally different results.
And that's really the beautiful thing here. These 2 processes aren't competing. They're like 2 sides of the same coin of life. You need both. Mitosis makes sure that you, the individual, can grow and stay healthy, and meiosis makes sure that your species can continue, adapt, and thrive.
Both of these processes are so elegant, so precise, and so essential for everything we call life. But, it makes you wonder, doesn't it? Given how unbelievably complex this whole biological dance is, what happens when the process messes up? What happens when it goes wrong?